The most interesting thing we've discovered in the last year speaks to the central dogma of molecular biology. So the dogma is the thought that everybody hands down. When you learn biochemistry, the great professor says, this is what is true, this is what we know. What we know to be true is that a gene has a sequence. This specifies the sequence of the protein, which drives it to fold up in one particular way. And this is the structure of the protein. What we have found is that there may not be one structure of the protein, there may be many structures of the protein. So like the transformer toys that refold from a robot to a truck and back, these proteins can refold into different shapes. So one from the Ebola virus makes a butterfly-shaped dimer that it uses to drive itself around the cell to where it needs to go. It refolds into a ring that binds RNA, that's genetic material at the center, to control how the virus replicates in itself. It rearranges again to make the zigzagging long filament to build and release new viruses from the infected cell. So depending on what it needs, it reassembles itself. It would be like if the clothes in your closet, the threads unknitted from swimsuits to reknit themselves into cozy sweaters in the winter and then reknitted themselves into an umbrella when it was raining. We have never known proteins to do this before. This is magnificent. We see this in these viruses because these viruses have such small genomes. They have to do more with less. They only make a few genes. Instead of 20,000, they make 4. And so this discovery, which we made by using viruses and by using the structure of these proteins, now opens up this whole new area of biology. A fantasy of mine is well, it's not really a fantasy. I think it's probably true. I think that there are lots of proteins that can rearrange into lots of different structures instead of being, the old dogma of it makes one structure. And I think this can help us understand a lot about biology and disease. What if there was a cancer that was caused by the protein being stuck in the ring instead of a zigzagging filament? And we don't have the tools to know this, right? If we're screening for cancer, we look for, is the gene turned on or off? Well, in both cases, the gene is on. Is the protein present or absent? Well, in both cases, the protein is present. It's just different. It's the ring or it's the filament or they're different shapes. And so this is a whole wide new area of biology that could address everything about life itself on the molecular level. And we've just opened it up in my lab. And so there's lots to learn and lots to explore.